Hey everybody, welcome to our first lecture on the nervous system. Super complex system, going to coordinate all of the body's actions, going to process all the body's sensations, going to transmit signals to and from different parts of the body, and does so many other things as well. So many different things. It's an amazing system, and we're going to scratch the surface on it with this intro. So, first thing we're going to do is divide up our nervous system into two big parts, the central and the peripheral nervous system. So two big parts, central, peripheral nervous systems. Central nervous system is your brain and your spinal cord, well protected in your cranial cavity and in your vertebral cavity. Brain and spinal cord, central in terms of their position, central in terms of their importance. Central nervous system, CNS, processes sensory information, generates motor responses, and also connects those two, like integrates the information, figures out what's going on and how best to respond. Peripheral nervous system, on the other hand, is everything outside of the CNS. So it's all the nerves, and we see so many nerves here, that are connecting the body to the brain and the spinal cord. So the peripheral nervous system, in fact, we got two different kinds of nerves in it. We're going to have nerves that are coming out of the brain. Those are called cranial nerves. We're going to meet 12 pairs of cranial nerves later. We're also going to meet 31 pairs of spinal nerves that are taking information in and out of the spinal cord. So in our peripheral nervous system, we know we've got all these nerves outside the CNS. And there's two anatomical varieties, cranial and spinal. But then we can also divide up our peripheral nervous system in a more functional way. We can have nerves taking information into the CNS from the body. They would be considered sensory because we're taking sensory information into the CNS. They're also considered afferent because they're going towards the central nervous system. Afferent means carrying towards. Then we're going to have motor efferent nerves. These are nerves taking commands from the CNS to effectors. Our mnemonic device is the word same, sensory afferents, motor efferents. Here is a sensory afferent nerve cell taking information from receptors in the skin towards the spinal cord over here. Here is a motor efferent nerve cell taking commands from the central nervous system to an effector, which in this case happens to be muscle. And effectors are generally muscles or glands. Now we can further break these guys down. We can take these sensory afferent nerves and describe them as being either somatic or visceral, with somatic ones taking sensory information from skin, muscle, and joints to your CNS. The word somatic refers to the body. So these are all like bodily sensations, like what's touching the skin, what are the muscles doing, what are the joints doing. Visceral sensory afferent nerves are, on the other hand, taking information from blood vessels, so regarding blood pressure, regarding blood pH, blood gas content, or taking information from organs. Like if you take a deep breath, you can tell your lungs are stretched. If your stomach is full, you're aware. If your bladder is full, you're aware. Those are visceral sensory nerves doing that. So two different varieties in our sensory afferent division. Similarly, we can divide up our motor efferent division. This is almost an identical division, just different names. It's very similar though. So first we have the somatic nervous system. This is going to send commands from the CNS to skeletal muscles. And this is generally voluntary. Not 100% voluntary, but generally voluntary. Then we got our autonomic nervous system. Our autonomic nervous system is going to send commands from the CNS, but to smooth muscle, cardiac muscle, and glands. These are things that are generally involuntary. So we control the skeletal muscles that move our body, they're voluntary. That's the somatic nervous system. We don't control smooth muscle, cardiac muscle, and glands. That's involuntary. And that's under the effects of the autonomic nervous system. And then the final breakdown we do is, before we go on to the final breakdown, here are our effectors here. So these are skeletal muscles here: triceps brachii, latissimus dorsi, deltoid. You're going to learn all these. Glute max. You're going to learn all these guys. Occipitalis. Anyway, these are all skeletal muscles pictured here under the command of your somatic nervous system. Smooth muscle in the urinary bladder, cardiac muscle in the heart, a gland like the prostate gland, all under control of your autonomic nervous system, which is involuntary. All right, the final breakdown we can do is we can take that autonomic nervous system and then separate it into two parts. A sympathetic branch called the sympathetic nervous system, then a parasympathetic nervous system. So remember, autonomic was going to cardiac muscle, smooth muscle, and glands. And basically, these two systems describe the situations in which you're going to turn those different systems on. So if you're in a fight or flight situation, a acute stress situation, it's your sympathetic nervous system. Rest and digest situation, it's your parasympathetic nervous system. Now, we're actually going to spend a ton of time this semester on the nervous system. We're doing our intro. That's what this slide set is about. And then we're going to deal with the brain and the spinal cord and spinal nerves. Then we'll deal with cranial nerves. We'll deal with the autonomic nervous system way down the road. So we're going to come back to this in great detail. Don't worry. All right. 3 basic jobs of the nervous system. We've already hinted at them. The nervous system has to take sensory information in. It has to integrate or process that information. Then take commands. It's called motor output. Sending commands to effectors. So 3 basic jobs: sensory input, integration, motor output. 2 basic cells in my nervous systems. Neurons, and I have 1 neuron in this picture. This is the body of the neuron right here. There's his nucleus. These are the extensions that we'll talk about later. But this is a neuron. And then supporting the neuron are glial cells. All these little nuclei around here are glial cells. Neurons are going to do those 3 basic jobs: sensory input, integration, and motor output. Glial cells, which are also just known as glia, like you take the L off that, they are my supporting cells for my nervous system. And we'll see what we mean by that word support. Okay, so we're going to do the glial cells first. We have 6 kinds of glial cells. All of them are supporting the nervous system in some fashion. We have 4 varieties of glial cells in our CNS and 2 varieties of glial cells in our PNS. My 4 CNS ones, and we're going to go through all of these guys, are my astrocytes, the microglia, the oligodendrocytes, and the ependymal cells. Now, on the other hand, the 2 types of peripheral nervous system glial cells are going to be my satellite cells as well as my neurolemocytes. All right, so let's go through these 6. In this picture here, I see my 4 CNS glial cells. Here's a microglial cell right here. Notice it's many arms. Those arms are going to be crucial for its function. Okay? This is an astrocyte, also with many arms. This is an oligodendrocyte. Look how the oligodendrocyte is covering. It's got these arms that's sticking out. Everybody's got arms so far. It's got these arms sticking out, covering up parts of neurons. Then finally, I got my ependymal cells up here. And notice my ependymal cells. These things here are actually cilia. Ependymal cells are going to have cilia. Okay, let's go through these four one at a time. Astrocytes, big jobs for them. Number one, they regulate the interstitial fluid. So the blue here is the tissue fluid. In this case, it's in the brain, but there would be tissue fluid in the spinal cord as well. And the contents of that tissue fluid, like the homeostasis of that tissue fluid, making sure it has the right levels of ions, for example. That is the purview of the astrocyte. Astro means star, cyte means cell, so kind of in reference to the shape. Astrocytes also help form something called the blood-brain barrier. And we see the astrocyte here is interacting with this tiny blood vessel, this capillary here. Basically, the blood-brain barrier, and we'll expand on that in a second, is going to protect my CNS cells from whatever bad things, toxins, pathogens, etc., that could be in the blood. So here is a blood capillary. This is the lumen of it, where it says luminal. This is where I'd find blood in this capillary right here. And what I've got here are these four green things. And these four green things they're the feet of the astrocyte. And basically the astrocyte is putting these feet on the blood vessel and kind of covering it up. And this is creating a barrier between the blood and the brain's tissue fluid. So if there are bad things in the blood, it is going to be hard for them to sneak their way out. So two big jobs for astrocytes: blood-brain barrier and regulate the tissue fluid, the ISF, for our central nervous system. Microglia. Here's my microglial cells. Microglia are phagocytic cells, hence all the arms. Arms are good for engulfing. These guys are going to remove infectious agents. These guys are going to remove damaged or underused neurons and synapses. Synapses are connections between neurons that we'll talk more about later on. Here I see a microglial cell. He's got these processes. He can use these processes to swallow microbes, cellular debris, whatever needs swallowing. So he's kind of like a macrophage. He's kind of like a dendritic cell. Imagine there was a little microbe, like, there we go, come on. Right here, there's a little microbe. What could happen is the two arms could join right here. And if the two arms join, we then engulf that microbe and we can destroy it, help basically clean up the tissue fluid of our CNS. So that's our microglial cell. Oligodendrocytes create this structure called the myelin sheath. The myelin sheath is an insulation. And it's an insulating and protecting layer on axons of my neurons. So here is a neuron, here is a neuron. I see a bunch of processes coming out of that neuron here. But then there's a process, an extension covered up by the oligodendrocyte. That extension is called an axon. Axons send electrical signals, And this axon under here, the oligodendrocyte, is, by covering it, is insulating it, like the covering on a wire. It's also providing it with some physical protection. So oligodendrocytes create this insulation. So this insulating layer right here, created by the oligodendrocyte, is known as the myelin sheath. Here I see an oligodendrocyte right here. I got a nice neuron right here. Several processes called dendrites that we'll meet later. Then another process called an axon. This axon sends these electrical signals, and we need to insulate that electricity carrying structure. So the oligodendrocyte is wrapping its arms around it here and here, also wrapping around this axon as well. And this covering created by the oligodendrocyte in the CNS here is called a myelin sheath. Okay, my final guy, my final CNS glial cell, this guy called the ependymal cells. The word ependymal refers to like a upper body garment, like a piece of clothing you wear in your upper body. So a covering, and something is going to be covered or lined by ependymal cells. Ependymal cells are going to line cavities within the brain. So this tannish region up here is actually a brain cavity. Brain cavities are called ventricles. Ventricles, we've got 4 big ventricles, and all 4 ventricles are lined by ependymal cells. The spinal cord has a cavity called the central canal, also lined by ependymal cells. And we mentioned this earlier, but the ependymal cells contain cilia. And what does cilia do? Of course, cilia sweep. And the sweeping action of the cilia here helps move the CSF, the cerebrospinal fluid that would be in this cavity here. I've got a brain that has been sliced frontally. And you see this space right here? This is called a lateral ventricle. That is the left lateral ventricle. This is the right lateral ventricle over here. And the 2 lateral ventricles, if we were to zoom in on the cells lining them right here, they would look like this. These are ependymal cells, and here are the cilia, and the cilia are going to sweep, and by sweeping, circulate the fluid in here, which is called cerebrospinal fluid. They help it move from ventricle to ventricle, also help it move out so it can go around the brain, and help it move down into the spinal cord as well. This is the spinal cord in cross section, and the spinal cord has a passageway right here called the central canal, and this guy is also lined by ependymal cells. Okay, so those are my 4 central nervous system glial cells. I've also got 2 peripheral nervous system glial cells. They are known as satellite cells. And then the other name I gave you was the neurolemocytes. And in this particular picture, the neurolemocytes are labeled as Schwann cells. That's their old-fashioned name. Okay, satellite cells, which I see here and here. Lots of satellite cells surrounding a neuron cell body. Lots of Schwann cells or neurolemocytes surrounding an axon. Okay, let's figure out what exactly these guys do. So my satellite cells are going to do something a lot like what the astrocytes did in the CNS. So the astrocytes had one of their big jobs was to maintain the tissue fluid, the ISF, around brain cells, spinal cord cells. So that's CNS ISF. Satellite cells are doing a similar thing, but now in the peripheral nervous system. So they're generally found very near the cell bodies of both sensory and motor neurons in our peripheral nervous system. They help maintain the tissue fluid around it, and they make sure the neuron doesn't get hungry. They provide it with nutrients. Here I have a zoom in on a region near the spinal cord. We're zooming in on something called the dorsal root ganglion, which we will actually meet later on. So we're zooming in on something called the dorsal root ganglion. So in this upper picture here, here's the spinal cord, and then right here is the dorsal root ganglion, which is a swelling near the spinal cord, and we're zooming in like right where that little square is. And in here, I've got cell bodies of sensory neurons, and then around them, I've got satellite cells, helping maintain the environment for these sensory neurons. Now, the neurolemmacytes, on the other hand, which are also known as Schwann cells, they form the myelin sheath that's going to be the insulating layer around axons of the peripheral nervous system. So remember, oligodendrocytes. We saw oligodendrocytes, right? Let's go back. Where are they? There's an oligodendrocyte forming the myelin sheath around a neuron's axon, but this is a CNS neuron, and his axon is being insulated. In the PNS, Schwann cells, or neurolemmacytes, I've got 6 different Schwann cells right here. Like, this is the way the Schwann cell wraps around. It's kind of like crazy. They like just coil themselves around the axon. And they're creating this insulating and protective layer around the peripheral nervous system axon. And this is what the Schwann cell or neurolemmacyte does. Neuro refers to the fact that we've got a neuron here. Lemmo means covering. So neuron covering cell. Pretty good name. And the myelin sheath is super important for proper conduction of electrical signals down that axon. Okay. With that, we're done with glial cells. We did all 6 kinds of glial cells. We are moving at a good pace here. So now we're going to talk about the neuron. We have probably, I don't know, like 100 billion neurons in the human body. And that is just a crazy number. Okay? Maybe not quite 100 billion. Maybe something more like 86 billion. Anyway, I totally just double-checked that number as I'm doing this lecture. Real-time fact-checking going on. Anyway, that's just a ridiculous number of neurons. And this is, we have big brains. We are super smart. And our big brains and our incredible smartness is because of this large number of neurons. Now, this right here is a typical neuron, and it's got a major portion, the cell body. By the way, the cell body is also known as the neuron's soma. The word soma literally means body. And then coming out of that soma, we have processes. And I see multiple processes known as dendrites, and then a single process down here known as an axon. So this is typical of a neuron, to have a cell body and have multiple dendrites, and then a single solitary axon. All right, so let's figure out what the cell body does and what these processes, these dendrites and this axon do. So the cell body of the neuron, as we said, it is this region right here. So basically, like, this is the cell body, and also called the soma. It is the place where we got the nucleus. Okay, we got the nucleus. We have a lot of organelles. So it is going to do like basic biological functions like biosynthesis, making the things the neuron needs. The soma is going to receive signals, so other neurons might come up and let's get rid of everything here. Another neuron might come up and have one of its processes hit on the soma to stimulate the soma. Not only do we have a nucleus in here, we have protein fibers called neurofibrils that give the neuron its shape. We have an ER. Remember what ER stands for? ER stands for endoplasmic reticulum. This is going to take me a long time to write. Endoplasmic reticulum, which is where we make proteins and lipids. The ER of the neuron is also known as the Nissl body. How do you guys feel about when we name things after people? I don't know if I quite like that. It's also called the chromophilic substance, the tigroid substance. I don't know if I like those. I don't know if I like that any better. Anyway, we have the body here. It's going to have my organelles. It's going to make things. It's going to receive signals. It's got my nucleus. It's got my ER, endoplasmic reticulum, which is also known as the Nissl body. And it's got neurofibrils, which are fibrous proteins in here that give it shape and structure. Okay. Fantastic. Oh, come on, arrow. There we go. Most of my somata are found in my brain and my spinal cord. Most of my somata are in my brain and spinal cord. So why do we have them there? Well, they're very well protected there. And of course, they're interacting there. They're forming networks there. So we think about all the things our brain and spinal cord are capable of. It's no surprise that most of our neurons' major parts, the cell bodies, are in the brain spinal cord. And when I say vast majority, we're talking probably 90% in the brain and the spinal cord. Another way of saying in the brain and spinal cord, of course, is to say in the CNS. Okay. We just said, like a moment ago, that the neuron has a nucleus. We know a nucleus is the part of the neuron or part of any cell where we find the chromosomes, the DNA, right? Well, we're also going to use the word nucleus in a totally different way. The word nucleus also refers to a group of somata. Somata is the plural of soma. So a group of cell bodies in the brain. So this would be a nucleus right here. So we'll talk about nuclei in the brain. Like, for example, there's a nucleus in your brain called the lentiform nucleus. And if I said there's a lentiform nucleus in the brain, I'm using the word nucleus not to describe a part of an individual cell where I've got DNA, but I'm referring to a group, like a huge group of neuron cell bodies. All right, so always fun when we use words in multiple ways. We also find groups of somata, groups of neuron cell bodies outside the CNS. So here's the spinal cord. I see nerve fibers coming out of it. Got some vertebrae here, by the way. Very nice. This right here is a collection of cell bodies, a collection of somata, and a collection of somata outside the CNS is called a ganglion. We looked at a ganglion. This was a ganglion right here. Do you remember when we did this? We looked at this because we wanted to see the satellite cells. Well, check out what I got here. Cell body, cell body, cell body, cell body, cell body, cell body. I got a lot of neuron cell bodies, a lot of somata, and a collection of somata is called a ganglion. So we were actually looking at this ganglion right here in that histology pic. And I got some other ganglia right here. I got a bunch of ganglia in this picture. How many ganglia do I have? 2, 4, 6, 8 ganglia in this picture. And a ganglion is a collection of neuron somata in the PNS, so outside the CNS. A collection of somata inside the brain is known as a nucleus. All right, good stuff. So that takes care of the soma. Let's now talk about the processes, starting with the dendrites. The word dendrite means tree branches. And dendrites branch like crazy. The picture doesn't do justice to the branching. A neuron might have 10,000 dendrites on them. Dendrites, the job of dendrites is to receive signals from other neurons or from sensory organs. So dendrites will receive signals from other neighboring neurons. So another neuron would have part of it come up and release chemicals, and that neuron would stimulate this dendrite. But then when the dendrite receives signals, the dendrite has got to send its own electrical signals down towards the soma, down towards, I can't draw dots very well, can I? Down towards the axon so the neuron can respond. The signals we find in a dendrite are referred to as graded potentials. More on them later. Okay, while a neuron has many branching dendrites, it only has a single axon. The axon is going to take electrical signals away from the cell body. So in the dendrites, electrical signals went towards the cell body. So in dendrites, electrical signals are going towards the cell body. In the axon, electrical signals are going away from the cell body. In the dendrites, the electrical signals were known as graded potentials. In the axon, we have electrical signals known as action potentials. Axon, action potentials. When the electrical signal gets to the end of the axon, and I see several axon terminals here, these axon terminals release chemical signals, which are called neurotransmitters. So the chemical signals released by an axon are called neurotransmitters, and those neurotransmitters can then stimulate another neuron or an effector cell, like a muscle cell or a gland cell. Okay, so those are my trio of parts to a neuron: the soma, the dendrites, and the axon. Let's zoom in on the axon and learn about its parts. So the axon has many parts. The conical beginning of the axon is known as the axon hillock. So basically, like this whole part of the axon right here is called the axon hillock. Hillock is another one of those words that's like just not common, not a common English word. Like a hillock is a small hill, but I don't know who uses that word. Like you wouldn't be like, look at that hillock over there. Anyway, the axon hillock is right here. I know what you're thinking, that it's like there's another line going into the axon hillock. Well, the axolemma is actually the cell membrane of the entire axon. So I could point anywhere to the axon, anywhere that's not covered. So anywhere where I can see the yellowish cell membrane of the axon, that cell membrane is called the axolemma. Notice that for the majority of this axon, the axolemma is covered up by Schwann cells. Remember, the Schwann cell is also known as a neurolemmocyte. And you learned that neurolemmocyte meant neuron, covering, cell. So lemma here, like lemmo here, means covering. So axolemma is the covering of the axon. In other words, it is the plasma membrane of the axon. So the hillock is the beginning of the axon, the part connected to the cell body. It is conical in shape. The entire axon has a plasma membrane called the axolemma. Most of it is covered up by these bluish cells in this pic. These bluish cells are Schwann cells, aka neurolemmocytes. Now, the outside of a neurolemmocyte is called the neurolemma. Gotta love the vocabulary. The outer surface of a neurolemmocyte. The outer surface of a neurolemmocyte, also known as a Schwann cell. So the outer surface of a Schwann cell, so the outside, if I was like to touch the outside, I'd be touching the neurolemma. So the neurolemma is part of the neurolemmocyte. The neurolemmocyte covers the axolemma. Okay? All right. Now also notice that the Schwann cells leave a gap between them. There's a space between Schwann cells where I can see the axon and the axolemma. That space is called a node of Ranvier. A node. All right, sorry, I had to pause my video in order to have a drink, so I didn't like just cough like crazy. Like 40 minutes in, start to need to take a break. Anyway, we're going to finish this, and then we'll keep going. So node of Ranvier is this gap between Schwann cells. If you don't like the name node of Ranvier, it is also called a myelin sheath gap. With the myelin sheath, that's some terrible handwriting right there. With the myelin sheath being the covering created by the Schwann cells, or neurolemmocytes. It's got some other names as well, but this is probably the only other good one. Anyway, nodes of Ranvier are going to be crucial for letting the electrical signal move down the axon quickly. As we're going to see, the electrical signal, it's almost going to appear as if it leaps from node to node to node to node. It doesn't actually jump from node to node to node, but it appears as if it does so. Now, finally, the last part we have in this pic is something we mentioned earlier, the axon terminals, They are, actually, I got one other thing, don't I? I'm covering something up. I almost forgot. Let's do the telodendria first. So the axon comes down, 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 down. Then the axon has branches here. Branches at the end. That's what the word telodendria means. It means branches at the end. Telo means end. Dendria means branches. So branches at the end of the axon are called telodendria. Then every telodendrion is going to have an axon terminal, the little part right here. The axon terminals, notice they've been labeled as the secretory region because that is where my neuron releases its chemical signals. And those chemical signals, I'm going to abbreviate NTs for neurotransmitters. So when an electrical signal, remember that was called an action potential, goes down the axon and goes down quickly because of the nodes of Ranvier, when the action potential gets to the end here, it releases, the ends rather, the axon terminals release neurotransmitters, which can then stimulate a neighboring neuron or a neighboring effector cell, like a muscle cell or a gland cell. All right, we're going to end this first video in just a second. Let me just do one more thing. I got one other neuron here. Here's its soma. Here's its dendrites. Here's its axon hillock. Here's the axon. This axon has some big branches. So we had branches at the end. The branches at the end we just did, those were telodendria. When there are big branches, the big branches are called axon collaterals. Axon collaterals let a single neuron get to more effectors, or let a single neuron get to more other neurons. All right, with that, we will end our first nervous system lecture. We're going to pick up in the next one talking about synapses. This is a solid 42 minutes right here. Really good. We're like a third of the way through, I think. That's awesome. All right, as always, come talk to me if you have questions. See ya.